So we have talked about some of the anchor proteins in passing. Now let's look at a more detailed view of these anchor proteins, how they interact with each other, how they interact with the cytoplasmic domain of integrin molecules, and how they link the cytoskeleton indirectly with the beta subunit of integrin molecules. Here is a detailed view. Here you can see that there are two subunits of integrin, the beta subunit and the alpha subunit. In this case, the binding is shown to a collagen fiber. We have said that earlier that while the alpha subunit of integrins seems to be more responsible for extracellular ligand binding, however, the beta subunit is basically the cytoplasmic domain, but rather the beta subunit is more involved in interacting with the regulatory proteins inside the cell. When integrins are in resting state, alpha and beta subunit, the cytoplasmic domain of these two subunits are in close proximity and they're sort of entangled with each other, which constrains the integrin in low affinity conformation. So unless these two tails are slightly dissociated, they will not be able to bind their ligand very effectively. So dissociation of the two tails signals that tell this integrin to dissociate these two cells, basically result in binding of integrin to the ECM substrate. The long cytoplasmic protein here shown here in yellow is talin, is the key regulator of integrin affinity. Tallins bind the integrin beta tail. We are going to call the cytoplasmic domain as the tail. So tallins bind the beta tail, and causing this binding basically causes the separation of two tails, alpha and beta tails. They separate, and this results in relieving of the constraint and activating the integrin. Talin is composed of an N-terminal globular domain and an extended or a rod-shaped domain, which is followed by a motif, a region which can bind actin filaments directly. So this part is going to bind the beta tail. There's this rod domain along which different other anchoring molecules can bind. However, at the very tip, there is a domain of tallin where actin filaments can bind directly with this protein. Tallin is ubiquitous cytosolic protein. It is found in almost all cells, and it is found in high concentrations in focal adhesions. It is capable of linking integrins to actin cytoskeleton directly or indirectly by interacting with vinculin, or for example, alpha-actinin can also do that. Here we have just shown vinculin. So here is the direct interaction of vinculin, direct interaction with actin filaments, and also indirect through vinculin in this case. So here, this talin interaction with beta tail is mediated by PIP kinase. You might remember that we talked about phosphoinositol phosphate, phosphatidylinositol bisphosphate. So this basically kinase modifies this interaction of talin with PIP kinase, basically modifies talin and permits it to interact with the beta tail. Additionally, I have to tell you that PIP kinase is activated by another protein which is not shown here, but I would like to mention it. It's the SRC protein, abbreviated as SRC. This protein is in turn activated by FAK, focal adhesion kinase. SRC is basically short for sarcoma. It is a proto-oncogene. It is a non-receptor tyrosine kinase that is encoded in humans by SRC gene. This phosphorylation of the SRC gene or activation basically results or happens by FAK, focal adhesion kinase. We'll talk about focal adhesion kinase in the next module, but basically FAK activates SRC. SRC basically activates PIP kinase. PIP kinase basically results in activation of PIP kinase results in activation of talin domain, this yellow ball-like domain, and in this activated domain, this domain is activated, it can bind the beta tail of the integrin molecule. If the PIP kinase and SRC, these two molecules, are expressed in a cell synergistically, it induces anchorage-independent growth of non-malignant cells. Malignant cells do not rely on attachment to a substrate. They can divide without attachment to a substrate. But most cells, normal cells, which are other than, for example, blood cells, they need to be attached to substrate in order to divide. So if we express SRC and PIP kinase in a constitutively active form, they basically result in cell division, which is independent of integrin's binding to its ligand or the extracellular matrix. Here you can see there's another complex, ARP2-3 complex. Basically, it stands for actin-related protein. It can basically form modular hybrid complexes containing an actin nucleating core. I've said that for adhesion molecules, the cytoplasmic domain of adhesion molecules can also serve as a seed from which actin filaments can grow. Here is one such seed, which is composed of ARP2-3. It is also interacting with vinculin. This results in growth of actin filaments from this seed out into the cytoplasm. So this is basically some of these. We have seen some of these anchor proteins and how these anchor protein interaction with the cytoplasmic domain of the beta subunit basically results in activation, not only activation of integrin molecules, but also most importantly, they link cytoskeleton with the beta tail of the integrin molecule.